Now that you understand the structure of DNA, we need to get into the actions of DNA. And one of the actions it has to take is replication, or as it is entitled here, DNA semi-conservative replication. Think back to what you learned earlier about cells and the cell cycle, and you know that cells divide. They go through mitosis or meiosis. What you also learned at that time is that during interphase, and more specifically during the S portion of interphase, all the chromosomes have to replicate within the cell. They all have to make a spare copy because, of course, eventually one cell is going to divide into two, and so there needs to be an entire spare set of chromosomes so that each daughter cell receives a full and complete copy of all the chromosomal or hereditary material. That process during the S portion of interphase is, in fact, DNA semi-conservative replication. It can be a fairly complicated process, but one that is very important to be done accurately. I'm going to boil it down to an oversimplified level here to start off, in which I'm going to break replication down into just two steps. Both of those steps are operated by enzymes, and if you'll recall from earlier in our course, enzymes are chemicals that help to catalyze chemical reactions. So you can see in our two-step process, the first step is authored by an enzyme called DNA helicase, and the second by a different enzyme called DNA polymerase. The first step, DNA helicase, separates the molecule into two templates by breaking the hydrogen bonds. Now think about that statement for a second. You know that DNA is a double helix. It is a two-sided molecule. And you know that they are connected, those two sides, in the middle of the quote-unquote ladder by hydrogen bonds between the adenines and thymines and hydrogen bonds between the cytosines and guanines. So if you look at the picture over here to the left, here is our double helix, and this is just going to be, and I think the phrase that is typically used would be unzipped right down the middle. I'm going to go back and forth between our pictures here for a second. Here's a different representation. You can see here is our DNA double helix. Here is the helicase enzyme, and it is simply going right down the middle of the DNA and breaking the bonds between the adenines and thymines and the cytosines and the guanines. So here is an adenine-thymine combination between that, and that bond has been broken. Here was a guanine cytosine that was broken, and so the helicase is just moving from right to left in this case, and it is breaking those bonds between the two bases. That gives us two what are called templates. Here is one template. Here is the other template. Let's go back for a second and look at step number 2. Again, another enzyme, this time DNA polymerase, adds free-floating DNA nucleotides to each template. So here is a DNA polymerase enzyme. Here, working on the other template, is another, and you can see what it's doing. Here, in this case, it is taking a cytosine, free-floating cytosine nucleotide, and adding it to the guanine. So cytosine to guanine. Previously, it had added an adenine to this thymine, and this thymine to this adenine. So it is just working its way down that particular template, adding free-floating DNA nucleotides. The same thing is happening down here on this template. It added a C to this G, it added an A to this T, it added a T to this A, and it is currently about to add a free-floating adenine to this particular thymine that's on the template. Because of those base pairing rules, because an A and a T always go together, and because a C and a G always go together, what you will eventually end up with is an identical strand here as compared to here. Look at the two. TNA, TNA. A and T, A and T. Same thing here and here, CG, CG. You end up with an exact replica. This also brings up the need to explain the name. DNA semi-conservative replication. Semi, as in semicircle. Semi means half. Conservative means to keep. So if you think that eventually this is going to be one strand and this is going to be another, here you have the old original DNA strand. Here you have new. Here you have the old original DNA strand. Here you have new. So within this totally new DNA, you have one half that is old or has been semi-conserved and one half that is new. Same thing up here, one half that is old or semi-conserved and one half that is new. Here are some videos that you can watch that deal with the process. And here is a slightly more complicated understanding of DNA replication. There are things called Okazaki fragments and other enzymes that are involved in the process, like ligase. But as long as you know DNA helicase and DNA polymerase, I think we're off to a pretty decent understanding.